The topic of lecture 16A is protein and cell interaction with materials. Biomedical engineers are interested in these aspects because they are the basis for successful new tissue formation around implanted materials and devices. This slide, which we saw earlier in the course, is a reminder that at the injured site of a tissue, the process of wound healing takes place when the two sites, which are composed of similar or the same biological fluids, proteins and cells and extracellular matrix components, work during the process of normal wound healing to bridge over this particular gap and re-establish the continuity of the original tissue. The situation is totally different because an interface is formed when a synthetic device composed of materials is introduced as an implant in the human body. That interface separates the synthetic world from the biological physiological world, which of course contains cells, extracellular matrix, and biological fluids. Under these conditions, biomedical engineers encounter a major challenge. In order to have successful wound healing and new tissue formation, the questions that biomedical engineers have encountered and have tried to answer are how to promote desirable and timely interactions of the surrounding cells and tissues with implanted biomaterials and devices, and in such a way that normal tissue is regenerated and the new tissue is functional as well. Going back to that particular slide with the interface between the two worlds, the biological and the one surrounding tissues, we notice that the major difference between the two sides across the interface is the extracellular matrix. In the biological tissues, the extracellular matrix is now replaced in the tissues around implants by the biomaterials, the ceramic, metal, plastic, and respective composites, which are used to put together the implantable device. Information and inspiration on how biomedical engineers could achieve this ultimate goal, namely the formation of new and functional tissue around implant materials and devices, that information and inspiration came from lessons that we learned from advances in biology and physiology. To start with, the characteristics of the native extracellular matrix had to be studied and understood. The extracellular matrix of tissues is an environment that has many functions. It supports the cells contained in the tissue, but at the same time, because it contains a lot of bioactive chemical compounds such as growth factors, it provides that kind of stimulation needed for the function of the cells pertinent to new tissue formation. This multifunctional environment provides not only chemical but also biophysical stimuli. It's a dynamic environment, and related to specific tissues, there are biophysical stimuli which support and maintain the tissues and the organs of the body. For the purposes of this lecture, I will remind you of an important fact that we discussed earlier in the course, namely that proteins mediate the interactions of cells with substrates and promote the cell functions which are pertinent to new tissue formation. Some of those functions include cell adhesion, migration, proliferation, etc. This, of course, now explains why the title of this lecture is Proteins and Cells, because the chemical components of the extracellular matrix are, for example, proteins such as collagen, fibronectin, laminin, and other proteins which are specific to specialized tissues, proteoglycans, which are a form of proteins, and some examples of proteoglycans are aggregan, decorin, syndecan, etc., and glycosaminoglycans such as heparan sulfate, chondroitin sulfate, hyaluronic acid, etc. The list on this slide provides additional names of proteins and proteoglycans from various tissues. A protein which is found in almost all tissues is collagen, although it's different type of collagen. And for example, we encounter type 1 collagen in bone, skin, tendons. Type 2 collagen is the predominant type in cartilage. Type 4 collagen is found in the basal lamina, which is a component of the basement membrane, and so on and so forth. Our interest in proteins is because of a well-known fact that proteins mediate the interaction of cells on the surfaces of materials. For this reason, I will review some of the functions of the cells which are important for their survival and functions pertinent to new tissue formation, and bring to your attention the appropriate mediation of proteins. My discussion will be a brief one because all of you have either taken or are taking a course given in the BME department at UTSA, which is pertinent to cellular engineering, and I am sure you cover these aspects in greater details in that course. Cells have many characteristic functions. Some of them are listed on this slide. First and foremost, adhesion. Cells adhere on substrates and they adhere to each other to form the cohesive tissue. The morphology of the cells is pertinent and differs from tissue to tissue. Survival of cells is important. And in addition, we have apoptosis, which is what we called a programmed cell death. Motility, sometimes referred to as apoptotaxis, especially if it takes place on the surface of a material. Definitely proliferation. Differentiation, if that function is applicable, and it is, of course, in the case of stem cells. And then synthesis of macromolecules, which result in the formation of an extracellular matrix and other functions. Starting with cell adhesion, there are two types of that function. Cell substratum, which links the cells to the extracellular matrix, and cell to cell, which links cells to molecules on the membrane surfaces of other cells and results in the cohesiveness which we associate with the various tissues of the human body. Examples which are involved in cell-cell and cell-substratum interactions in biology include embryogenesis, angiogenesis, formation and growth of organs, cell migration and invasion, cancer metastasis, etc. Because of time limitations, in this course, we're gonna review only the adhesion of cells to substrates. For that purpose, we need to start with a definition, and that is of anchorage dependent. Those are the cells which require a substrate, either natural or synthetic, to first and foremost adhere. Cell adhesion of anchorage-dependent cells is the first interaction of these cells on substrates, and it is necessary for the survival of anchorage-dependent cells and their subsequent normal function. This interaction, cell adhesion on material substrates, occurs within 1.5 to 2 hours maximum from the initial contact of cells with substrates. This is the information that we have received as a result of using culture cells in the laboratories, a widely used experimental technique. Examples of anchorage-dependent cells, in this case they are osteoblasts, which are the bone-forming cells, is shown on this particular slide. This is light microscopy. Remember, all cells in the body have no color, with the exception, of course, of the red blood cells. Therefore, in order to visualize cells under the microscope, you either have to stain them, and that means that you have to fix them or to expose them to an alcohol or some other chemical compound which kills the cells, or if you want the cells to remain alive, you use techniques which try to play with the various shades of gray. As you can see in this particular slide, you can see the perimeter of the cell, and in the center you see a darker dot or oval, which is the nucleus of the cells. With light microscopy, this is how far you can go in terms of seeing details of cells. You need more powerful microscopy techniques in order to be able to see the cytoplasmic organelles. Examples of anchorage-dependent cells include endothelial cells, smooth muscle cells, osteoblasts, chondrocytes, fibroblasts, keratinocytes. You can see now a list of cells from many tissues. Endothelial cells from the vascular tissue, including, of course, smooth muscle cells, osteoblasts from bone, chondrocytes from cartilage, fibroblasts ubiquitous from many tissues, keratinocytes from skin, etc. How do proteins mediate the interactions of cells with substrates? In order to answer that question, we must take a closer look at cell adhesion on substrates. In other words, we need to investigate the mechanism used by cells when they adhere on the surfaces of substrates. The very important aspect which we must always keep in mind is that cells do not interact with bare material surfaces. Cells, in order to adhere on material surfaces, need the mediation of adsorbed proteins. The type, the distribution, and the conformation of the proteins will subsequently dictate on whether the mammalian cells adhere and they don't adhere, whether they survive or not, and whether they function, and therefore the process gives us the formation of new tissue. As mentioned earlier in this course, implantation of a biomaterial or device in the human body exposes them to biological fluids and tissues which contain many proteins. In addition, within seconds from the introduction of biomaterials and devices inside the human body, these are covered with adsorbed proteins. This process is illustrated on this slide in a cartoon fashion. You can see the selective proteins indicated by the yellow squares are the ones which are adsorbed on the surface of the biomaterial. Why is this particular step important? Because this particular step dictates the subsequent interaction of cells. The cells which now interact with the adsorbed proteins survive and function, while the other cells who were not lucky enough to find an adsorbed protein on the biomaterial substrate, they are not going to survive. In this respect, we have to keep in mind that the type, concentration, distribution, and conformation of the adsorbed protein are the characteristics, the parameters which dictate the subsequent adhesive interactions of the cells with the adsorbed proteins, pre-adsorbed proteins, I should say, on the material surfaces. The mechanism by which proteins mediate cell interactions with substrates have been studied extensively. They are summarized on the cartoon of this particular slide. What we have in the figure on the left is a substrate on which one cell, in a cartoon fashion again, rests. We have in the middle an oval insert which indicates the nucleus of the cells. If we now take the interface between the cell and the substrate and magnify that particular portion, what we see is the following. If we start from the side of the substrate, we have obviously a metal, bulk metal. On the top we have a layer which is the metal oxide. As you know, when metals are exposed to an environment which contains oxygen, such as the air which we breathe, the metals form an oxide layer. However, in biological fluids, there is some concentration of dissolved oxygen as well. Therefore, when a metal implant goes inside the body, there is always a metal oxide layer formed onto the surface of the bulk metals. In the figure on the right, we have on the top of the metal oxide a liquid portion, and the little Mickey Mouse faces there stand for water molecules, and that separates now the surface from the cell membrane, which is illustrated here in the old-fashioned way of a lipid bilayer. Mediating the interaction of the cell membrane and the cell, therefore, with the substrate is this serpentine notion of a green protein. The details of the mechanism have been studied and they will be discussed in the subsequent slides. What has been established, and it's well known now, is the following aspect of the mechanism of cell adhesion on substrates. Specifically, that there are so-called receptors, which are proteins on the cell membranes, which recognize and bind on specific domains, areas, regions, epitopes of the extracellular matrix proteins. These particular receptors, sometimes they're always available, sometimes they're hidden and they need to be activated in order to be exposed on the surface membrane of the cells, and some others have been activated and deactivated as a result of exposure of the cells to various environments. Example of such an interaction are the so-called integrin receptors on the cell membrane. These receptors recognize and bind on a 3-amino acid region, arginine, glycine, aspartic acid. If we use the single letter notation for these amino acids, RGD, which is the domain on the extracellular matrix proteins such as fibronectin, collagen, etc. The important thing to remember here is that integrins are the receptors on the cell membranes, and the RGD is the part, a domain, an area, an epitope on several extracellular matrix proteins with which now the cell receptors adhere, recognize, and then adhere the RGD sequence of peptides. With this slide, I would like to bring to your attention the following fact. Not all cells have integrin receptors, and not all cells, even if they have the integrin receptors, recognize only the arginine glycine aspartic acid sequence. When we have a group of a few amino acids, we call that a peptide. The peptide sequence, therefore, RGD. Different cells may have integrin receptors with which they recognize other sequences of amino acids, other peptides. The arginine glycine aspartic acid peptide was the first one to be identified for its adhesive properties when researchers worked with fibronectin. But later on, research revealed that other extracellular matrix proteins, such as collagen, vitronectin, laminin, thrombospondin, etc., have the RGD peptide. Therefore, many cells may recognize that particular sequence in these proteins and adhere with their appropriate receptors on that particular sequence. In addition, the bulky big molecules, which are proteins, may have more than one peptide sequence which is recognized by cells as adhesive. For example, see here on the first row, laminin is known to have the sequence arginine glycine aspartic acid. But in addition, laminin has a sequence which is composed of tyrosine, isoleucine, glycine, serine, arginine. That peptide is recognized by other integrin receptors, not only the one type with which cells recognize and adhere to the RGD epitope. Last but not least, certain cells require not only integrin receptors, but some other type of receptors. An example here is heparan sulfate proteoglycans. These particular receptors on the cell membranes, they recognize other domains in the extracellular matrix proteins. We call those domains heparin binding domains. One particular sequence, which is the amino acid sequence, which becomes now the adhesive domain or epitope on various extracellular matrix protein, is the sequence lysine, arginine, serine, arginine. With the one-letter designation, this sequence is KRSR. As a result of extensive research in this area, information regarding what cell membrane receptors of your favorite cells are used in order to recognize a particular domain in an adhesive protein of the extracellular matrix, all that information may be found in the literature, already investigated and published. If you happen to work with some very, what I may call, exotic cells, and such information is not available in the literature, you have to do the respective work yourself in the laboratory, identify the receptors on the cells, identify the domains on the extracellular matrix proteins which those receptors recognize, and therefore establish the details of the mechanism regarding how your favorite cell interact with the pertinent proteins in the extracellular matrix of their respective tissue. I shall add one more detail regarding terminology. The arginine glycine aspartic acid, or KRSR, all of these epitopes or domains are part of extracellular matrix proteins. Those proteins many times are referred as ligands, L-I-G-A-N-D. An updated version of the detailed mechanism, which now incorporates the information regarding the membrane receptors and the domains of the proteins, is shown on this cartoon slide. You can see now that we have on the upper part the interior part of the cell. In the middle, in this khaki colored double line, is the membrane. You see the integrin receptors. Integrin receptors, by the way, have two parts designated by alpha and beta, and there are different alphas and different beta and combinations which make a long list of integrin receptors on the various cells. In the remaining of the cartoon, you see now that we have again the green serpentine notions of the proteins of the extracellular matrix, but the cells now interact via their integrin receptors, and as you notice, they interact only with those domains which are their recognizable ligand. In this case, in red is the RGD peptide. Availability of cell cultures and development of appropriate techniques to maintain and grow cells in the laboratory made it possible to investigate the morphology of cells which interact with the surfaces of materials which in the slides that I'm going to show you are pertinent to implants in bone. Keep in mind that when the cells are in suspension as a result of the surface tension, they become very spherical. When they start interacting with a substrate, as shown here with one osteoblast, the bone-forming cell from rats, you can see the cell, this is within half an hour of the initiation of that interaction. The cell is still very, very, very spherical, but it has already made contact with the substrate by extending these finger-like projections and sometimes wider interactions of the cytoplasm with the substrate. Within an hour, however, you can see the difference now. The cytoplasmic extension has become much more broader, but in the middle of the cell you see this particular bump, which of course is the nucleus and the various other organelles of the cells. It will take about another half to 1 more hour to see this particular cell interacting completely and fully with the substrate. The following slide illustrates this particular case. In this slide, we see the morphology of rat osteoblasts adhering on bone from cows. This bone was devitalized, which means that it was treated in such a way that there is no indication or remnants of cells from bones. It's only the extracellular matrix. You can see the cell half an hour after the initiation of this adhesive interaction, within 1 hour, and definitely after 2 hours, you can see how flat the cell is, but it's still recognizable because in the middle we have this bump, which again is the indication of the nucleus and the organelles of the cell, the organelles inside the cell cytoplasm. The important thing to notice here is that if a cell is interacting with a protein which is pre-adsorbed on the surface of a substrate, the cell is going to become very flat and spread out on that particular substrate. This particular process takes place within an hour and a half, maximum 2 hours in the laboratory. If the cells do not interact with a particular substrate in the presence of whatever proteins you are testing, that means that the cell will never interact. As a matter of fact, if the cells remain non-adherent for an extended period of time in terms of hours, they're not going to survive, they're going to die. And that is happening within less than 24 hours of the exposure of cells onto substrates, physiological or naturally occurring, or the substrates composed of synthetic materials. The previous slides of cells adhering on substrates were taken using scanning electron microscopy. We have in addition fluorescent microscopy, and if you take the appropriate stain, and in this case is a stain called rhodamine phalloidin, which stains the F-actin filaments of the cytoskeleton, you can see when the cells are still making their initial interaction with the substrate and they are very, very spherical, that particular stain is diffused almost uniformly throughout the cell. And that photograph was taken within half an hour after the initial interaction of the stained cells with the substrate. However, after 24 hours, the cells have adhered now, they're alive and doing well. You can see how spread the cell is. You can see in the middle the round nucleus, and you can see this extension of the cytoplasmic finger-like projections with which the cell adheres on the substrate. Another way to visualize an adhering cell is shown on the cover of the Materials Research Society bulletin. Here we have a fluorescent micrograph of an adhering cell. You see the nucleus stained in blue. You see the F-actin filaments of the cytoskeleton in green lines. That is the so-called linear arrangement. And at the end of all these lines, you see little dots which are either yellow or red or both. Those are called the focal adhesion points. And it is something to keep in mind that the cells, when they adhere in the substrate, they don't make 100% contact. They adhere literally with the tips of the cytoskeleton, which are these so-called focal adhesion proteins. Extensive research in this field has revealed what is the composition in terms of proteins which are found at the focal adhesion points. This is very elegant research, and the results are very important. They have provided important information, and they're absolutely magnificent to know all the details of how a single cell adheres on a substrate via the mediation of adhesive proteins. An additional aspect which is very important for the adhering cells is the fact that through the receptors in the cell membrane, here the integrins alpha and beta shown along the cell membrane of this cartoon, via these integrins, and the fact that the cell now makes contact with the extracellular matrix component, here in a cartoon fashion fibronectin, which has been pre-absorbed on the substrate at the bottom of this figure. Via this particular mediation, what we know now is that signals from the outside world are transmitted inside the cytoplasm of the cells. And research over the years has revealed that what are those proteins which are composing the cytoskeleton of the cell. Remember, the cells do not have a skeleton, but they have a series of proteins which, depending on the morphology of the cells, the environment to which the cells are exposed, they make connections, and that particular line of connection is transmitted all the way to the nucleus. Here you see the beginning of that particular connection, and you have, let's say, tallin and vinculin and alpha-actinin interacting with actinin and who knows what other proteins inside the cytoskeleton to bring the message from the outside world inside the cell. Of course, later on, the cells bring messages from the nucleus via similar pathways to the outside world, and the cell, of course, functions in that way. The nucleus contains the programmed information which is pertinent to cell function. Signals and stimuli are needed to bring messages from the outside world to the nucleus, and from the nucleus to the other parts of the cell, which are now getting the message and bring about the outcome of the cell functions which are needed, for example, for synthesis and deposition of extracellular matrix, new tissue formation, proliferation, etc. Research regarding the transmission of those signals from the outside world to the nucleus, the outside world indicated on the upper left hand corner of this cartoon, the cell nucleus in the lower left hand corner of the cartoon. The transduction of the mechanism, the transmission of this mechanism has been the subject of extensive research by cell biologists, molecular biologists, biochemists, and colleagues in many other specializations of the basic sciences. This gives you an impression of some of these particular pathways which have been investigated and elucidated. It is an absolutely fascinating area of science. It has revealed a lot of good information which we can, all of us, use in order to bring about applications in the biomedical field. This is the end of lecture 16A with the title Protein and Cell Interactions with Materials.